right, so our next speaker is Tatenda. I'm not even going to attempt his last name because I'm going to butcher it. And Tatenda is a PhD student working with Dr. Mike Lee. And his project is really interesting because it's a collaboration between Iowa State and CIMMYT. And CIMMYT is down in Mexico, and it's the International Maize and Wheat Improvement Center. And so Tatenda is going to show us his research, and I just want to point out that one of the great things about Tatenda is his enthusiastic contribution to our spectacular hallway parties. He always shows up, and he's always excited to participate. So with that, Tatenda, the floor is yours.
Well, thank you very much for the introduction, Jamie. I'm missing my hallway parties, but they'll come back very soon. So as mentioned by Jamie, my name is Tatenda, and I'm a PhD student with Dr. Michael Lee. Today, I'll be giving a general overview of all the research that I've been conducting since I came to Iowa. And the title for my presentation today is the genetic assessment of maize populations selected for drought tolerance. So I'll start off my presentation just basically talking about where I am from. I come from Harare, which is the capital city of Zimbabwe. I grew up in Harare, attended the University of Zimbabwe in Harare, and did a bachelor's in crop science as well as an honors in crop science. From there onwards, I went to South Africa, the University of KwaZulu-Natal, where I did a master's in plant breeding. And then I worked for a small seed company called Capstone Seeds and got the privilege to work around Southern Africa before I came to Iowa State, 8,797 miles away from home. So I'll start off by mentioning the contribution of maize to global diets. Excuse me. So maize is a staple in a lot of countries in Africa as well as Central America, where it contributes very high in terms of in terms of calorific intake in those parts of the world. And most maize produced around the world is used for stock feed as well as biofuels, but it's important to note that 13% of all the maize produced goes to human consumption. And in areas where that 13% goes to, droughts normally are a common feature that cause a lot of problems with regards to the production of the crop. So one challenge we have, as I'm working with drought, one challenge we have is defining what a drought is. So for drought being a phenomenon that is well known across the world, it is interesting to note that there's no one widely accepted definition for what a drought is. And the Oxford English Dictionary states that a drought is a long period of time when there is little or no rain. Most definitions, depending on your disciplinary perspectives, drought can have different definitions. Well, as crop scientists, we're mainly interested in what an agricultural drought is, and that has been defined by Kulik as the difference between plant water demand and available soil water. It is important to note that between 1980 and 2020, in the United States alone, drought has resulted in crop losses with 250 billion. And for this study, I came up with my own definition of what a drought is. What is my drought treatment for this study, which I defined as the withdrawal of irrigation during the flowering period of the life cycle of a maize plant. So I'll mention the impact of drought on maize productions globally. The figure on the right, as you can see, I extracted from Science magazine. They had a special report in April, which was specifically targeting drought. And this alt showed different regions of the world where precipitation and soil moisture are their lowest or highest, and I have circled in red parts of the world where most maize production takes place. And it is interesting that in these areas circled, droughts are a common feature. For example, in this region, which is where I'm from, Southern and Eastern Africa, the 1992-93 drought resulted in a loss of over 70% of expected yields. And the drought in 2012 in the United States resulted in a 27% loss in terms of grain yield, expected grain yield. So fluctuations in terms of yield as a result, in terms of maize yield as a result of drought differ from year to year as well as geographic location. And globally, it's been estimated that 7% potential maize yields are lost as a result of drought. But as I've mentioned, as I've stated earlier, it all depends with what part of the region it is. And in some regions, the losses are quite higher than in certain parts of the world. So here I've extracted a figure from a publication by, I think it's a well-read publication by Cooper et al. And in this figure, it just explains the conundrum faced by plant breeders as well as farmers in breeding for as well as adopting drought-tolerant cultivars. So they did several experiments at different locations, and locations that were defined as drought susceptible. You find out that in this experiment, 2 hybrids were used, one which is drought sensitive and one drought tolerant. And under drought conditions, the drought tolerant hybrid gave a higher yield, which is expected. But compared to the drought sensitive hybrid, it generally would give lower yields under drought. This is expected, but as the conditions change towards being favorable, now we get to see a reversal of the situation where the drought tolerant hybrid gives less yield than the drought sensitive hybrid. Maybe this figure doesn't clearly show how important this difference is, but to a farmer growing, say, hundreds of hectares, that difference here can result in a farmer making it or not. And farmers really don't want to forego this yield penalty as a result of growing drought tolerant hybrids. So how have breeders been tackling this issue? So one of the issues with regards to drought tolerant is that in 2012, we had 2% maize acreage in the United States of America grown under hybrids marketed as drought tolerant. But by 2016, the number had shot up to 22% in terms of acreage in the United States that is grown under drought tolerant hybrids. What has caused this change? Well, there's a possibility that the 2012 drought could have made farmers want to grow more drought tolerant hybrids, and companies have responded after this drought, especially in the United States of America, where seed companies, for example, Corteva is their Aquamax line of hybrids that are conventionally developed to be drought tolerant, whereas Monsanto, which is now Bayer, developed a transgenic drought line known as the drought hybrids, known as Drought Guard. The figure on the right here shows the difference between a transgenic and a non-transgenic hybrid. For example, hybrid 1, we have a similar hybrid. The only difference between these 2 hybrids is one has got that transgenic trait, whereas the one is non-transgenic. And these hybrids were all grown under drought conditions, and you can clearly see how better performing the transgenic hybrids have been. So these transgenic hybrids contain the Bacillus subtilis cold shock protein, and several experiments, according to their data, have shown that adding this transgenic Bacillus subtilis cold shock protein B has improved stress tolerance. And it's not only for drought. It has also been mentioned that for cold tolerance as well as heat tolerance, these hybrids perform much better. So I move on to my study, where I'm working on drought tolerance on 2 maize populations. One of the maize populations is named La Posta Sequia, whilst the other one is maybe not so creatively named drought tolerant population. The drought tolerant population has been developed from superior performing genotypes or populations that have been brought together to create an initial cycle, and from that initial cycle, development of further cycles has taken place, whereas the La Poste-Sequia population was developed from elite, broadly adapted germplasm. And this germplasm was sourced from different parts of the world, from tropical West Africa, Central America, as well as in Asia. And here's just an illustration of the development of these populations. These populations have been developed in a period of over 3 decades by CIMMYT, CIMMYT Mexico, CIMMYT Zimbabwe, and CIMMYT Kenya. And for the drought-tolerant population, we have 9 cycles available. And from cycle 0 to 3, they were created using half-seed recurrent selection. And the selection was conducted under well-watered conditions. Whereas from cycle 4 to 9, we had full seed recurrent selection in the development of these populations, and they were evaluated, and the selection was conducted under drought conditions. Whereas for the here, population evaluated under drought all the way from cycle 0 to 7, using a full seed recurrent selection. So one might be interested to know, why did I pick these 2 populations to work with? So these populations have been widely used in terms of different studies that are related to drought tolerance. A lot of inbred lines and materials developed from, sourced from these populations have gone on to make some very successful hybrids, especially in subtropical and tropical regions of the world. So I was motivated to pick these 2 populations to work with in my drought studies. So from some of these studies that I've mentioned earlier, that I pasted in the previous slides, I managed to source some data from 2 publications, one by Monovu, 2005, and Grigory Edmidis in 1999. And these studies show that genetic gain has been made in these populations. For example, under drought-tolerant population, from cycle 0 all the way to cycle 6, from 3 cycles, basically, cycle 0, cycle 3, and cycle 6, the authors found that there was genetic gain starting from the earlier cycle to the later cycle under drought conditions. And the same can be said for La Poste-Sequia populations under drought conditions. It is important to note that all these studies were on per se performance of the cycles. So the cycles were tested as they are. There were no crosses or anything done. So, but genetic gain was observed after conducting these studies. So what is my study all about? My study, the main objective in my study is to assess genetic gains and phenotypic changes due to selection across these maize cycles that have been created, as well as evaluating genotypic changes across the cycles. And as I'm collaborating with CIMMYT, one of the key goals is to obtain or come up with donor lines for drought tolerance that are to be distributed across the world to those who are interested in using these lines in their breeding projects. So I structured my study into 2 parts. On the first part, in each cycle, we managed to create doubled haploid lines. So for example, under drought tolerant population in cycle 0, we pick promising individuals, and these individuals were used to create doubled haploid lines. The same was done for the La Poste-Sequia populations. And the second part of the study involves sampling individuals from the cycles. So by individuals, I mean sampling members of the population, that is the open pollinating individuals, and I would also use them for some of my experiments. So getting into my study, one of the main objectives in the first part of my study, that is the phenotypic evaluation of doubled haploid top crosses, is we're going to evaluate and compare genetic gains across the cycles under the 2 treatments, that is the well-watered and drought conditions. So in this study, it is important to note that we are using doubled haploid top crosses. So this is our doubled haploid cross to a common tester. We seek to evaluate responses of flowering time, plant height, and other traits. So a selection has been taking place, what has been happening to the other traits. The main focus in developing these populations was grain yield, but have we seen any other changes across time as we were selecting for drought? And how did I create the germplasm? So as I had stated earlier, we created doubled haploid lines. So for example, for the drought tolerant population, we had doubled haploid lines. And from each cycle, we managed to get differing numbers of doubled haploids. The same can be said for the Laposta Sequia. We had differing numbers of doubled haploids obtained from the different cycles. For the drought tolerant population, I had 5 cycles. So for the both population, we picked cycle 0, which is the original cycle. And then I went on to pick odd numbered cycles to do my experiments on. And we crossed each and every one of the doubled haploids with a common tester, that is CML 550. CML 550 is a CIMIT inbred line that is drought tolerant. Why did we pick this inbred line, one might ask? And we picked it because it is drought tolerant, as I've mentioned, and it is also a good general combiner. By general combiner, I mean it will cross well with the inbred lines from different heterotic groupings. And because we're not so sure which heterotic groupings these inbreds belong to, CML 550 was a good option for us. And we ended up creating our doubled haploid crosses. And these are the ones that have undergone phenotypic evaluation. And these are my test sites. This is where we did our phenotypic evaluation. So I had all of them were in Western Mexico. I had one location in Los Mochis, that is in the Sinaloa state, one in Nayarit state. There was one site in Coyantes, that is in Oaxaca. The pins keep moving into the ocean, but I can guarantee you we planted these plants on land. And I also had one site that is at Tlautizapan, which is a CIMIT research station. And for the drought tolerant population, named DTP, we did all our experiments over 3 years. And for the Laposta Sequia population, all our experiments were conducted in 2 years. So what constitutes a drought treatment in this study? As I had stated in the definitions, defining drought is a bit tricky. And for us, we decided that we would stop irrigation at the V12 stage, at the middle to end of the V12 stage. And V12 basically is a development stage in a maize plant where we have 12 leaf collars visible on a maize plant. And we withdrew irrigation all the way across this tasseling stage or the flowering stage, and only restored our irrigation at the R2 stage, which is generally called the blister stage. So we picked this stage because, as you can see from this figure, if you withdraw irrigation at this stage, a maize plant stands to lose the highest amount of possible grain yield. So we were trying to come up with a severe drought. That's why we picked this stage. And for all our trials, we planted in December. And the reason why we planted in December is in these western parts of Mexico, if you plant in December, there's a high chance that no rainfall will occur in the growing season, or limited rainfall will occur in the growing season of a maize plant. So our evaluations were we had an alpha lattice design with 2 replications per entry. That is for our drought treatments. And for our well water treatments, we only had 1 replication, and had special designs with a plant population of 66,000 plants per hectare. We used 66,000 plants per hectare for our trials because in the regions where we did our trials, most farmers use that plant population. I mean, it does vary, but 66,000 is a common plant population used by farmers in these parts of the world. And since we have doubled haploid lines, we decided to genotype all our doubled haploid lines. And the reason I have an objective to assess haplotype frequencies across the 2 populations. And the definition of haplotype tends to vary as well, just like the definition of drought. But one common thing is that these are generally alleles that are inherited as a unit in most cases. And what as part of the objectives, I seek to identify regions that may have undergone selections, as well as investigate if there's convergence between the GTP and La Poste-Sequia populations. So as we are breeding for drought in these two populations, are these populations moving far apart from each other, genetically speaking, or are they becoming more similar as a result of breeding for drought tolerance? So this summed up basically the study on my double haploid lines. And as we were conducting these experiments, I realized that we do have individuals from the original cycles where we created our double haploid lines. And I decided, well, why not use them to conduct a similar experiment? And that's what we did. And we started phenotyping cycles from top crosses. So these are your open pollinating cycles. And these cycles we crossed to the same tester as we did with the double haploid lines. And we were hoping to evaluate the effect of recurrent selection on these populations. Have these populations been improved, so as to say? I mean, ideally, we hope that they've been improved in terms of they are now giving the later populations have higher grain yield compared to the previous ones, but no experiment has yet been done to test these individuals as top crosses to see if any advances have been made in terms of improvement. So in terms of our phenotypic evaluation, we basically sampled 4 cycles from the DTP population, as well as sampled three cycles from the La Poste-Sequia population. As we did with the double haploid top crosses, we used CMO 550 as well, and we bulked seed within each cycle. So what we planted now for the trials was bulked seed from each and every cycle, because we came up with this idea along the way as we were doing the double haploid experiments. We only started our trials in 2020, and this time we only had 3 locations in 2020, and we also planted this December. So I still have the material on the ground growing in 3 states of Mexico, that is Morales, Sinaloa, and Jalisco province. And the final part of this study was to do basically genotypic assessments of these cycles that we have sampled from the population. So we're seeking to identify regions that have undergone selective sweeps. So whenever there's favorable mutations in a population or we conduct selection, we get certain parts of certain alleles or blocks of haplotypes that are favored, and regions surrounding these favored alleles or mutations tend to reduce in terms of genetic variation, and they normally leave signatures. So we want to see if there have been any selective sweeps in our populations, and we want to see if the patterns of change in haplotype frequencies are similar in the 2 populations. If the population's been changing in terms of cycle in the same manner or not as a result of selecting for drought. And a comparative analysis with the double haploid study is one of the main reasons why we decided to work on these cycles, because as we created these double haploids, we are not sure if there was some form of bias. By bias, I mean certain individuals might be more inducible as we create double haploids. So there's the possibility that we might have a biased sample of the cycles. And if we now sample the cycles as they are, not necessarily double haploids, we might have a more representative, a more representation of the populations. And we sampled each of the cycles, as I've mentioned, and we're going to conduct genomic scans per cycle to determine those regions that have substantial changes in terms of haplotype frequencies. So that basically sums up all the 4 parts of my study, and for now I have conducted all my DH top cross study, and we're working on that data, and we still have the cycles under being grown in the 3 locations in Mexico, as I had stated earlier. So with that, I would like to acknowledge the people I've worked with on my project. That is my major professor, Dr. Lee, I have Dr. Somak Dutta in the statistics department. He's been helpful with the analysis of my phenotype data. Dr. Matthew Afford for the ideas he's given me in terms of the genotypic analysis, as well as at CIMIT. I have Dr. Terry Mona here, Dr. Tanda Liwayo, as well as Dr. Sarah Hen, and also the technicians I've worked with whenever I've gone to Mexico to conduct my study. Here, I just added a picture during lunchtime as we were harvesting our crop. With that, I would like to thank you for listening, and I'll be willing to take up any questions.
Well, Tatenda, I'll start with a question.
Yes.
I loved your description of how the population was developed, but what were the founders of the population? Is it a biparental cross? Is it more like a NAM population? As we've discussed, this is not my area of expertise, so I was just looking for some more details.
So depending on the population, for the drought-tolerant populations, there were programs running across the world where, especially in Mexico and Southern Africa, where breeders had already started working on. So those inbred lines were collected from these parts of the region. For example, with the drought-tolerant population, they had 200 sources come in, and those 200 sources were screened. And after screening, the 13 most promising ones were combined now to create an original cycle, that is cycle 0. And for the drought-tolerant population, they used individuals that were already that had high yield, that were high yielding under drought conditions, but not necessarily under well-watered conditions. And the opposite was done for the La Posta Sequia population. I don't know if that answers the question, Jamie.
Sure does, thanks.
Tatenda, I assume that during the recurrent selection, they use the test cross, right? Do they use the same testers or different testers? So was that, done, side by side or separate programs?
So the programs were separate, and for each cycle they did use testers, but they didn't use one tester. They used several different. I think it was 3 or 4 testers, and this was done over a period of 3 decades. So different breeders came in along the way, and there were slight modifications along the way as they created these populations. So they've been developed basically in a period of almost 2 decades.
Mm-hmm. But the table you showed about the per se performance, it seems like there's still per se performance. There was a under well water condition and drought, they were tracking each other, right, General?
Yes. Sorry, can you come up back? Can you mention that? I didn't hear you well.
The per se performance under well water condition versus the drought condition, yes. They're generally tracking each other for those different cycles, right?
Doc, I'm not quite getting the question, or maybe.
Oh, they're correlate. The per se performance under the drought condition and a well water condition. They were correlated somehow.
Oh yes. Oh yes. And with some of the results that I've also found, because I'm now doing the same experiments but using top crosses, I found similar correlations or patterns. So when they do well when a cycle does well under well-watered conditions, it seems to take up the same pattern under drought conditions, even though the yields differ. That under the well-watered would generally have a higher yield, but I'm finding a highly correlated pattern with regards to that. Yes.
Have you found any lines where that yield gap has been erased between drought and well-watered conditions?
Oh,
that's the goal, right?
Yeah, that's the goal. So from the results that I have found so far, there's still a bit of a gap. I don't know. Maybe I should show. This is just part of some of the results that I found for grain yield. I don't know if it's quite visible. You can see that for the La Posta Sequia, it's red. You can still see that the gap still exists, but the pattern, it's like what Dr. Yue did asked, seems the same in terms of response, but the gap still is there with regards to.
That's
but
fascinating.
ideally, we're trying to work to get to a point where the gap is a bit lower. That's the whole goal, yeah.
Hi, Tatenda. This is Chi.
Hi, Chi.
Very nice structure of your project. I enjoyed that. So since you're showing the slides, I'm curious about this plant height and year height. There seems to be a going down trend. Is that something associated with drought selection?
I would not necessarily say associated with drought selection, but I would suspect that breeders would be purposely selecting for lower plant height and ear height, because remember, we started off with a general with a population of a mix of different hybrids, and the initial selections were conducted around in the 90s. And there's been a trend of, especially with machine harvesting, people tend to want shorter maize hybrids with shorter ear height. So this might be an intentional, I'm not sure about it, but this is what I would suspect, because from some papers I've read, there's actually a positive correlation actually where we've been hitting taller plants actually yielding much better than the shorter ones. But this trend I find interesting that, and if you see under cycle, this reduction seems to start at around cycle 5 or 7. So that's why I'm suspecting this might have been purposeful breeding to look for shorter hybrids or shorter lines.
Yeah, that makes sense. Thanks.
You're welcome.
Other student questions? Okay, well, I would vote that we thank our speakers again very much for their participation today. And anybody who wants to stay on with additional questions, stick around. And there's usually a smaller group where maybe it's not quite as intimidating to get your questions answered. Thanks, guys.